Imagine yourself being put in the hospital because you are dying of a heart disease, and the only way to survive is to get an exact clone of your heart. The only way to do this would be through cloning, which would give you an exact copy made by your organs. The discovery of cloning is one of the most exciting yet controversial topics today. According to the article, The Benefits of Human Cloning from humancloning.org, published in 2012, it states that a heart attack could be reversed from human cloning technology by cloning healthy heart cells and injecting them into the damaged areas of the heart. The speech will include the history of cloning, how it was discovered, the 3 different types of cloning, and the ethical issues related to cloning. First, let's copy our way into the history of cloning. According to the history of cloning from Lean Genetics, last updated July 18, 2015, it states that in 1996, the first mammal, which was a sheep named Dolly, was created by somatic cell nuclear transfer. Ian Wilmot and Keith Campbell were the discoverers of this. An example would be 2 owls. You would take a cell out of this owl and a cell out of this owl, and then you would fuse them together using electrical fusion, and then it would create an embryo, and then you would put the embryo into a new mother, and then it would create a baby owl, which it would be like the sheep. Dolly would be an example, but I used owls as references instead. According to the history of cloning from Lean Genetics, last updated July 18, 2015, it states that in 2013, the human embryonic stem cell was created from somatic cell nuclear transfer. To do this, they took skin cells from a patient and fused it with donated cell eggs. Now that we have talked about the history and discovery of cloning, we are going to move into the 3 main types of cloning. The first type is gene cloning. According to the article titled Cloning from the National Human Genome Research Institute, last updated on June 11, 2015, it states that gene cloning is done by inserting a gene from one organism into the genetic material of a carrier, also called a vector. Some examples of this are bacteria, viruses, and yeast cells. The second type of cloning is reproductive cloning. According to the article titled Cloning from the National Human Genome Research Institute, last updated on June 11, 2015, it states that reproductive cloning is done by removing a mature cell, like a skin cell for example, from an animal and copying it and transferring the DNA into a nucleus that has already been removed. It should be noted that when it is transferred, it is not the exact identical, like the animal or the human isn't the exact as the clone. The last and final type is therapeutic cloning. Therapeutic cloning, according to the article titled Cloning from the National Human Genome Research Institute, last updated on June 11, 2015, states that therapeutic cloning involves creating a cloned embryo just to produce embryonic stem cells with the same DNA as the donor cells. This is used to help understand diseases and to also discover some treatments for the diseases. Lastly, let's duplicate our way into the ethical issues associated with cloning. There are both pros and cons to both animal and human cloning. One pro is that infertility could officially be eliminated. According to the article, Pros and Cons of Human Cloning from Health Research Funding, last updated on December 6, 2013, it states that being able to take cloned cells to create a younger twin of the mother or father would allow infertile couples to officially enjoy a family without having to go through the stressful process of being infertile. A con is that it reduces the sense of individuality because a clone is a twin, so a lot of people argue that you don't get a new person, you just get the same person, which doesn't really make sense. Another con is the possibility of faster aging. According to the Pros and Cons of Human Cloning from Health Research Funding, last updated on December 6, 2013, it states that premature aging issues can even cause premature death as a result of older cells being used to make a clone. Clearly, there's a lot of history associated with the cloning of both animals and human. It is an outstanding topic, because there's so much to it. There's even 3 types of different cloning. It is also causing a lot of controversy as if it is morally right or not. So, again, imagine yourself being in the hospital because of a serious heart condition and only being able to survive by getting an exact clone of it. Would you do it or would you not because you think it is not ethically correct? Thank you.